Okay, on to our next lipid, and these are phospholipids, which are extremely important in biological systems. So let me first draw a phospholipid. Very similar to a triglyceride, except for one major difference. So there is my fatty acid tail. Here is a second fatty acid tail. But instead of having a third tail here, we have a phosphate group. And this last oxygen could be bonded to another group. We'll just put an R there. So this new group here, this is a phosphate group. And this is polar. And you can see that it has a full negative charge, which makes it polar. It has a full negative charge. And then these guys here are the same things as before. Two non-polar fatty acid tails. So this is non-polar. We can also call these two sides different things. The polar part likes to interact with water because it's polar. So we call this hydrophilic, means water-loving. So this region is hydrophilic. But this region down here, the non-polar part, which won't mix with water, we call this hydrophobic. It's not afraid of water, it just won't mix with water. Now the fact that a phospholipid has both of these things makes it an excellent barrier. So it makes a good barrier. And this is exactly what it does in our bodies. This is the main component of our cell membranes. Main components of our cell membranes. Okay, so let me draw a cell here, and it has the phosphate group, its two fatty acid tails, and then it also has another thing sandwiched together, same thing. So phosphate group, two fatty acid tails, phosphate group, two fatty acid tails. And this runs along the entire cell. This isn't drawn to scale, but we have this throughout the entire covering of the cell. So this area outside the cell, there's some fluid. We'll call this the extracellular fluid. So fluid, you should think of liquids of water. And on the inside of the cell, we call this the intracellular fluid. So we would want polar things on this side and also polar things on this side. In the center, where the fatty acid tails are, this is a non-polar region. They want to get away from the water. Okay. So the polar phosphate group faces this fluid and also faces this fluid. Now the non-polar fatty acid tails are inside, not touching any liquid, are away from the fluids. So when an object wants to come into the cell or exit the cell, it has to bypass or has to pass through this membrane. Not everything can pass though. If something is too polar, if it's too large or too charged, it won't pass. So another way to say that it selectively allows things to come in and go we call this semi-permeable. Semi-permeable. And this lipid here, this is called a phospholipid. And since it's two of these layers sandwiched together, we call this a bi means two, a phospholipid bilayer. So be comfortable with explaining why this is called what it is. Okay, so that's it for lipids. Now on to amino acids and proteins. Let me first draw an amino acid. H2. Okay, so here's an amino acid. This group here, this is a carboxylic acid group. Carboxylic acid group. And acts just like an acid from before, can donate that proton. Now this group here, this is an amino group. So now if you take those names together, you have amino, and then this part of the acid group. This is why we call this guy an amino acid. So here is an amino acid. To make a protein, you have to take these amino acids and put them together. Not just one, not just two, not just three, but a bunch of these put together on the order of hundreds. That will make a protein. So let's draw the reaction of combining these amino acids. Okay, so let me, oh, I forgot to mention the R here. This depends on what amino acid you are. It changes depending on what the amino acid is. So the R group varies based on what amino acid you are, based on the amino acid. Okay, so back to that reaction. So here's my carboxylic acid group. Don't forget the lone pair is there. My amino group. Now let me draw another amino acid. So this is also the amino group, but just drawn out a bit more explicitly. There's that, and then here is my rest of the molecule. So in this reaction, this nitrogen's lone pair is going to attack this carbon right here. The electrons here are going to go to this oxygen, and then this lone pair is going to take one of these hydrogens, and while that happens, this lone pair is going to recede back to this nitrogen. So at the end of the day, a water is going to leave. So let's draw the product. NH2, CH, and then we have the carbon here, and here is this new bond that was just formed. Nitrogen got its lone pair back, still has one of its protons, and now we have the rest of the molecule. And then this is abbreviated COOH. And the other product here is water. So this is also a reaction of a dehydration because water is a product. Okay, so this new bond that formed, we call this a peptide bond. Peptide bond. That's a new bond form. And this new structure that we just created, we can call this a dipeptide because there are two peptides put together. To make a protein, however, it's not just one, again, not just two, not just three. It's a bunch of these on the order of hundreds put together with peptide bonds. There's one amino acid put together there, you know, a couple hundreds of these guys, many, many, many of them. Okay, now what do proteins do? I haven't explained anything about proteins. They do a few things. Let's take an example. Amylase is a protein. Amylase completes a function, right? So amylase would take starch and cut this up into glucose. So this protein did something. Another example of a protein is hemoglobin. Hemoglobin in blood, and it carries oxygen. Carries oxygen. So it didn't do anything, now it just kind of carries something. A third type of protein are structural proteins, like they make up a structure. So myosin is a structural protein of muscle. Structural protein. So proteins do a variety of things. Structural protein of muscle. Okay. All right. So now on to nucleic acids. Or maybe I'll save this for the next video.